One final video that really has an important theme for this course, and that's thinking about how we can embed things into the membranes, creating these different organelles to provide additional structures, additional functions, additional roles to these membranes. I want to remind you that if you have more surface area, more membranes, that also means you have more proteins that are embedded into those membranes. And since proteins are responsible for many, many different functions in the cell, that actually gives membranes functions too. These are just some examples of what proteins can look like when embedded into the membrane. We have proteins that are responsible for transport across those membranes. An example of this would be that nuclear pore, which is capable of allowing things in or out of the nucleus. But we're also going to see functions that include receptors. Receptors don't actually transport molecules, but they allow cells to communicate across one of those membranes, receiving a signal on one side and then producing a response on the other. We're going to look at two different organelles that have lots of membranes within them and proteins that are responsible for functions that these membranous organelles carry out. The first are mitochondria, which are the sites of cellular respiration, and this is going to be lecture 7, which we'll do next week. It's a metabolic process that uses oxygen to generate a lot of ATP. And then chloroplasts, which are found in plants and algae. They're the sites of photosynthesis, a metabolic process that creates organic compounds from carbon dioxide. So now I'd like to ask you, pause the video if you need more time, but what are the 4 basic components of a cell? What 4 things do all cells have? Well, if you think about the 4 things that every cell has, it starts with a DNA genome. If you take a look at mitochondria and chloroplasts, you're going to find a small circular DNA genome inside of each one of these organelles. That's consistent with a prokaryotic cell. They're also going to have a cytoplasm or a cytoplasmic-like fluid, although you're going to learn we don't call it a cytoplasm anymore because they're no longer individual cells. They're also going to have ribosomes because they're capable of synthesizing their own proteins. But those ribosomes are going to look a lot more like prokaryotic ribosomes than eukaryotic. And they're bounded by a membrane around the outside. These are membrane-bound organelles. These observations led to what's known as the endosymbiont theory. Endo meaning internal, symbiont, sim, like being together. It's an endosymbiont theory. The idea is that as an ancestral eukaryotic cell at some point engulfed a prokaryotic cell that was capable of doing cellular respiration. When it did so, it actually slowly and over time, that prokaryotic cell adapted to living inside of the eukaryote and started to simply carry out the function of cellular respiration, which benefited both the host and that bacterium. Eventually, it evolved to become what we know today as a mitochondria. It's thought that for some cells this happened again, except this time we engulfed a photosynthetic prokaryote, which gave rise to a chloroplast. It's important to know the structure of each one of these endosymbiotic organelles because we're going to be looking at this structure as we connect it to cellular respiration and photosynthesis. Mitochondria have 2 membranes. They have an outer membrane, a smooth outer membrane, and then an inner membrane, which folds into structures known as cristae. These cristae are important because these are what's going to create the large surface area that gives this endosymbiotic organelle the ability to do large amounts of cellular respiration. Between the membranes is a space known as the intermembrane space. And then inside of that inner membrane is the mitochondrial matrix, where the ribosomes and the circular DNA are found. This is an example, and you're going to see this slide again, where you can see that arrangement. There's an outer membrane, an intermembrane space between the outer and inner membrane. There are proteins embedded in the inner membrane that are responsible for cellular respiration, and then there's a mitochondrial matrix that fills inside. Chloroplasts are going to be very similar but arranged differently. They have two outer membranes, and then an internal membrane is arranged into little membranous sacs called thylakoids. So we still have a membrane inside that's responsible for creating that large surface area. These thylakoids are what contain the photosynthetic membranes. The internal fluid that surrounds the thylakoids is known as a stroma. Inside of the thylakoids, we call that a thylakoid space. And if you take a look at one of those thylakoid membranes, you'll see that they are studded and embedded with proteins necessary for photosynthesis. Now, so far, we've really focused on those intracellular components. Not only the arrangement of the membranes, but of the components within those membranes and the functions that they give them based on the structures that they form. But most cells synthesize and secrete materials outside of the plasma membrane. Right now is a fantastic opportunity to remind you that those components outside of the membrane were once inside of a vesicle. So material that's secreted outside of the cell is actually going to be produced in the lumen of the ER. And what you're going to see is that there's going to be a number of different glycoproteins that are synthesized by the rough ER, as well as glycolipids, which are lipids that contain sugar groups attached to them. All of these are extracellular, meaning outside of the cell. There are also intracellular components, which are suspended in that cytoplasm. This would include microfilaments of the cytoskeleton forming that cortex. These would be produced by smooth ribosomes. First, I want to show you the most common, or at least for you guys, the one you've seen and talked about the most on the outside of the cell, which is a cell wall. And yes, some eukaryotes have cell walls. Plants and fungi have cell walls produced from monosaccharides and polysaccharides. This includes chitin in fungi and cellulose in plants. And these are very important. They provide structure, and they're necessary for how these cells live. But animal cells, which includes you, do not have cell walls. Outside of your cells is what's known as an extracellular matrix. It's a network, a net that's actually formed that connects glycoproteins, not only those that are embedded into the membrane, the plasma membrane itself, but secreted to create this network of fibers like collagen. And then this anchoring of this network to proteins that are still embedded into the membrane. So this extracellular matrix does provide some level of structural support. It's also necessary for cells to adhere to other cells, which is very, very important in how cells regulate. You exist as tissues, which are cells touching other cells, being arranged into layers and boundaries. But this also allows cells to communicate between the inside and the outside. The proteins that span this membrane, called integral proteins or integrins, are capable of connecting the outside to the inside. So one of the critical structures of the extracellular matrix is collagen. Each of these proteins found outside of the cell has carbohydrates or sugars attached. So how would we refer to this sugar protein complex? Pause if you need more time. Good, this is a glycoprotein, named for the glycosidic linkage, which connects these saccharides, and the protein, which is the core structure of this glycoprotein. Which ribosomes would synthesize this glycoprotein? Yes, these are bound ribosomes. So this is going to be ribosomes attached to the rough ER. Now, we've already created and described 4 different functions of proteins. We started with the very first one we talked about were enzymatic proteins. Their function is to accelerate the rate of chemical reactions. We've also talked about structural proteins. This includes not only intermediate fibers like keratin, which is the example given here, but microfilaments and microtubules. And these structural proteins can interact with motor proteins like dynein or myosin. And these allow the cell to have movement. So motor proteins are going to be proteins that create a movement function. And then receptor proteins, which we more oftentimes call integrins. These are proteins embedded into membranes that receive signals on one side and then communicate them to the other side of that membrane. And we're going to have an entire lecture, lecture 10, where we talk more about this process of cell communication. Just a few final components that I want to introduce you to. One of them is cell interactions or cell junctions. There's more information about this in your textbook, and I would like you to read and understand that these are there, although I'm not going to ask you to know the different types of cell junctions for this class. These cell junctions, however, provide a really important component for this cell to be able to exist in tissues, which, as you know, is critical for you as a multicellular organism.
See fibronectin fibrils in the extracellular matrix of cultured cells. Images of cells expressing a fibronectin green fluorescent protein fusion were collected every 5 minutes. Notice how the adhesive fibrils can stretch considerably in response to cell movements. Credit: Harold P. Erickson, Duke University Medical School.
So this is an example of a tissue network that's formed. It doesn't look exactly like the tissues in your cell, but this shows you how these cells are touching each other, and it's necessary, it's important. Now, I want to leave you with a final theme here, and that is that even though we've spoken about each one of these organelles, as you're going through and studying and preparing for the next components of this class, including for exam 1, which is coming up very, very quickly, it's really important for you to start thinking about how each organelle connects to each other one. I strongly recommend that you create a list of all of the organelles and cellular components that we've talked about in this course, and then challenge yourself to randomly draw 2 of those from your pile and connect the 2 of them. Sometimes this is very easy, and sometimes it requires you to step back for a little bit, think about the larger context of those cells, and think about how each cell can go through and combine with another one. There are 5 final questions for you to try walking through and see if you understand them. Please make sure that you pause the video if you need more time to go through them, but these should be 5 questions that you try on your own before you hear my answer. So pause the video, try all 5, and come back if you need an explanation. So what structure is shown in this image? Well, even though you've seen this image before, hopefully you'll recognize that this is an endoplasmic reticulum. We're showing you both the smooth and the rough ER here, which is the only membranes studded by a ribosome, which immediately picks this out for you as an endoplasmic reticulum. What cell type is this? Well, this would be a plant cell. Not only can you see that there are chloroplasts here, chloroplasts being these elongated structures, these dark elongated structures here, but there's also a big vacuole filled with seemingly nothing in this image. That would be a central vacuole, which is necessary for plant cells. There's also a thick border around this cell, which is a cell wall made out of cellulose because this is a plant cell. Which cytoskeletal component is shown in red? In this case, the fact that these cytoskeletal components are around the outside of the cell tells us that this is the cell cortex, which is made out of microfilaments, and the protein monomer that forms microfilaments is actin. Remember that these components, both microfilaments and microtubules, can both polymerize and depolymerize, in part because they're made of monomers. so we can form and deform them as needed for the cell. And one example that demonstrates this importance is that the polymerization of these microfilaments gives rise to the ability for some cells to move via pseudopodia. Which of the following is not a protein? Even if you didn't know this, each one of these, A, B, and D, was proteins, hopefully you know that cellulose is made of carbohydrates. And so I will leave you with this. The question of, can you go through a cell and identify where these macromolecules are? Not only which macromolecule, but the function of those macromolecules, the enzymes that synthesize them, their structures, naming the bonds, and then connecting those macromolecules to other macromolecules. Here's your review for the things that you need to know, including microscopy, identifying microscopy and components of microscopy, the endomembrane system as a whole and the individual components, the vesicles that are formed both as components of that endomembrane system and vesicles that have specialized functions, the cytoskeletal components of that cell and how each one of those cytoskeletal components is composed, as well as the properties that it gives to a cell, and then taking a closer look at the outside of cells, both cell walls and extracellular matrices for cells that don't have cell walls. And there are some really fantastic review components that are found in your textbook that will allow you to walk through each one of these components in the cell, as well as view them as a larger component. So please make sure you take advantage of that textbook as you review.